with that, we are going to invite Dr. Bolus, Darren, Benjamin, you can stay up there. We'll be on stage for Q&A.
Yeah, and as we're getting ready for Q&A, just a bit of housekeeping. Thank you everyone for joining us here today. Just a couple things here is one, this is not a place for specific individualized medical advice, so this can be questions about general PSC, not about your personal health care. The other thing is we at PSC Partners value your privacy, and this is a public forum, so do not disclose information about participation in particular studies or clinical trials. We cannot have that information being shared during this. You can ask about the studies themselves, of course. And I'll be on the virtual Q&A as well, just to make sure those questions get brought up to the audience.
Okay. Well, I would like to talk, Ben, we didn't. Thank you for traveling all the way for the presentation. All right, we have a question there. Matt?
Yeah, I was curious about the problem where PSC might be multiple diseases wrapped up together and hiding under a trench coat. How do you take all the different trials and medications and figure out the difference between something not working and something working great for a subpopulation and not working that well for everybody?
Chris, do you want to address that?
Yeah, excuse me. Great question, Matt. This is really, I think there's two issues here that you're alluding to, and that has to do with these phase 2 studies. And how do you move something that's even a positive result into phase 3? And maybe Ben wants to answer that, because it's a real leap of faith that just because you see an improvement in alkaline phosphatase, ALF, whatever our markers are, that it's actually going to have an effect on outcomes, because we can't see that in those early trials. So number one is, if you have a positive result, how confident are you that it's actually going to work? So that's a limitation we have currently. And the other piece you alluded to was the heterogeneity of PSC is enormous, as Dr. Yamam was alluding to in the initial talk. And how do we know that, well, this drug didn't work in the total group of patients we studied in a clinical trial, which may not be representative of the whole, we know is not representative of the whole community of PSC, number one, because we have certain criteria to get into the studies. But is there a subpopulation that it might work in? And the trials are relatively small because PSC is a relatively rare disease. So out of 30 patients that might be in a treatment arm, how do you know there's not 10 that got a benefit? And I don't think we have an answer for that. I mean, that's why it's a great question. So I can talk a lot but not give you an answer. So I don't know if anyone has other.
Well, I was going to say, is there, to build on that, are there approaches in like classifying patients in certain groups, like, you know, large duct PSC versus small ducts, and how we design studies to see the benefit?
Yeah, and I think that maybe, Ben, you can, I don't know if you want to talk about this in terms of like who you're including in the Eloscope and why.
Yeah, I can try to add insight as well. I mean, I completely agree that because of the heterogeneity of PSC and still so many questions about, as we discussed earlier, what causes it and what makes a patient at risk for progression, it is challenging. And even for our study that we're soon to start for elafibranor in PSC, you know, we have to, as you probably picked up from my brief presentation, like patients with the more advanced disease, we're not including. So patients with like Child-Pugh B or C cirrhosis, decompensation, because there's a belief that potentially those patients' disease is too advanced to benefit from elafibranor. And then what I didn't share in detail, but if you look at our full inclusion-exclusion criteria, we also, in a sense, need to exclude patients who are so early in their disease and doing so well that in the course of a clinical trial would probably never have an outcome event anyhow. So, you know, we're trying to find that Goldilocks patient of, based on what we know about our drug, if you got into the study, there may be benefit that we can capture in 2-3 years' time. But by excluding those other patients, maybe we're missing an opportunity, and we would only know that if we studied those patients as well.
I think we'll take a virtual question, and then we'll get to you, Tom.
Yeah, so we've received a few virtual questions online. The first one I want to bring up is what is the current ongoing research in small duct PSC versus large duct? If there's been any recent developments over the last years, and if there's anyone you could point someone to for further research there in studying that versus large duct PSC?
So I think that the small duct PSC is a tough one. It is included in our natural history studies, our cohorts to understand the disease progression. Oftentimes they are excluded from some of the clinical trials for a variety of reasons, mainly because they tend to do better. And it's also can be a very even more heterogeneous group of patients that we call small duct. And I'd say that the number one thing about small duct PSC that we're learning is that if you have small duct PSC and you don't have inflammatory bowel disease or colitis, you need to have genetic testing to make sure you don't have a genetic cause of your small duct PSC. Even if you have PSC that's a little there's some MRCP changes, but you don't have IBD, you should probably get tested for that, because increasingly we're finding patients that have this PFIC, or at least are a gene for PFIC, that actually have this condition. So I'd say that's the number one thing we're learning most recently.
I guess the other piece is the small duct PSC patients don't progress, right, as much and with less complications. And I think it's good that you highlighted, we're including them in the natural history study, but the research, some of the research now is including, I think WIND includes small duct PSC. All right.
We do in KLID, though.
We are doing it on the KLID
And
study.
I think the registry also, right? The registry, yeah.
Okay. Tom, for a question.
So, yeah, we've been talking for some time, actually years, about this biomarker thing. I guess this is directed to Dr. Polis. And I guess for me as a patient, close to 20 years now dealing with this, what is the status of research on the causes of PSC, which would really enhance and speed, I think, the drug development and therapy development? I'd appreciate your comments on what is it that's causing this disease, what's causing it to progress.
So, yeah, so I'll separate what caused it, because this is where I think we as a research community have really failed. We've thought that if we understand the cause, we'll come up with a treatment and things like that. So 20 years ago, when we first started getting into this, the whole, the cutting edge research was in genome-wide association studies, these GWAS studies. We'll understand the genetic basis of the disease, and then we'll understand what causes it. So, and that was the thinking at the time. And so how do you do this? Well, you take a bunch of patients with the disease, PSC, and then you take a bunch of people without the disease, and you compare their genetics, and you say, oh, look, these 20 genes are different, and they're more prevalent in PSC, so, well, somehow they cause this disease. Well, yeah, we found genes. The most important gene was a gene we already knew about from the 1970s and 80s. So it didn't really add much. And there are a few others that have a little bit of effect, but they're also seen in other inflammatory and autoimmune diseases, so really not specific to PSC. And then when you take those genes and you say, okay, well, what about if we go look within patients with PSC, which of these genes are then associated with worse prognosis? We found one gene that could help explain why some people have worse outcomes than others. And nothing further on that. Nobody could understand why it was. The function of the gene wasn't known. So GWAS kind of didn't help us. So, okay, that goes. So what's the latest technology? What's the latest thing now? Well, the microbiome. So what are we doing now? Well, we're taking patients with PSC, comparing them to the healthy control, and maybe we'll throw in some IBD patients, and we'll compare their microbiomes. What have we learned? I would say not a whole lot. Yes, there are some microbes that are different in PSC compared to IBD, compared to healthy controls. Some of that may be due just because patients with PSC also have liver disease, so some of the changes you say are also from liver disease. So I think again, I think we're failing by trying to understand what causes PSC. What's more important is, what causes PSC to progress. And I think there's some sense of getting this, some of the metabolites, some of the microbes in the gut, they've been associated with this. I think those studies are very hard to do, though. We're completing a proteomic study looking at blood tests, and we've got some candidate genes there that are very interesting in certain pathways, and they tend to be very good at predicting patients that are kind of at the same point of disease, but kind of separate in terms of their outcomes. And so we're trying to validate some of that work, but it's really difficult to, it takes years, right, from the time you collect a sample to when someone has an event. And so we're going back to blood we collected 10, 15 years ago and testing it now to see what happened. And we're doing another study with some clinical trial data. We're trying to get this started from 2 prior clinical trials that were done, finished up 10 or 11 years ago, and then follow up with those patients, see how they're doing, and see if that data we collected previously is useful to predict those outcomes. So that's sort of where we're at with trying to develop biomarkers, but it's still some time away. And I'll also say, the FDA, in terms of using that as a biomarker, and I don't know, Ben or Darren, you can comment on this better, but my understanding is they're not going to accept a biomarker until there's already a treatment that shows that the biomarker changes and then you have better outcomes. So in PBC, they accept alkaline phosphatase because with URSO, alkaline phosphatase goes down and you get better outcomes. So and even there, it's not to the level of validation. They'll accept just alkaline phosphatase. So there's still a long way to go with biomarkers. I don't know.
We have a virtual question asking about the association, and I'll also add about the interactions between PSC and fatty liver or MASH.
So the question is the interaction between fatty liver and PSC. I don't think there is any direct connection per se unless they exist as a comorbid condition. Interestingly, we don't see it that often, but it's possible in someone with metabolic syndrome, including diabetes, obesity, hypertension, hyperlipidemia, they could have fatty liver inflammation from fat adding to the liver injury that's from PSC, possibly in combination can accelerate injury. But luckily, I don't see it that often. We see it a little bit more common in PBC, about like maybe 20-30% of our PBC patients, not such a common thing for PSC. One more question maybe from in-house? Yeah, go ahead.
Thank you very much, y'all. My name's Adam. I'm a patient. I'm interested in there's 2 things, so I guess maybe I'll just let you choose which one. I'm thinking about the Sweden study, and it was the Stokelin et al., the statin use associated with improved outcomes in patients. Just curious about that, your opinions on that, or what you think about the implications of that. The other one's a little bit different. It's to do more with like epigenetics and any connections that you all have heard about or are curious about between like ACEs or toxic childhood stress and later inflammatory disease, specifically PSC. So 2 different questions. You choose your own adventure.
So the question about statins and liver diseases, and not PSC specifically, has been investigated mainly through studies that are retrospective, meaning you look back at patients that have a disease and you look at those that got treated with a statin versus those that didn't, and you look and see, do these people survive longer, do better? And so with statin therapy for a number of liver diseases, it seems that there is people that get a statin do better than those that don't. The issue with those types of studies is that the people that get these drugs versus those that don't are not necessarily the same types, right? People that have more advanced disease, you're probably not going to start on a statin. You're going to be more concerned that if there's a liver toxicity, it's not going to go well. So you're kind of biasing the evaluation. And so we always have to take those sorts of studies with a grain of salt. And, you know, that's like a first level evidence. It makes us think about it. It makes the hypothesis plausible, but doesn't prove it. And so the Swedish study is then taking that and actually doing a clinical trial of rosuvastatin to look at this. And so that'll hopefully give us some more confidence or dispel the whole theory. There is also, though, a study the NIH is funding of rosuvastatin in cirrhosis. So there's enough evidence there that, and this is all comers, all patients with cirrhosis, regardless of cause. There's a handful maybe with PSC in there, to see if it actually works there. And I think that will also be quite helpful if it shows an effect there, that, you know, there'll be maybe more suggestion of using a statin. Regarding the epigenetics, not a lot in PSC. I will say the one thing that was kind of interesting was one of the trials looked at what's called methylation of DNA, which is, so epigenetics are changes to our DNA that are not inherited, but they're modifications of DNA that affect the genes that are expressed and proteins that are developed. And they can occur in utero or anytime during our development. And it turns out that this study was quite interesting. The PSC patients had this, what they call methylation clock, which kind of estimates your age. Were very advanced. I mean, it was unusually strange compared to other liver diseases and others. So, you know, it was interesting. Maybe there's something there, but as far as I know, not much more has come from that.
We're actually out of time. You have one quick question, maybe?
One
Okay.
quick one.
Okay.
This morning I heard sort of common kind of symptoms that you're measuring. One would be pruritus, and the other was fatigue. Now, when you're doing these studies, one of these sort of seems subjective, and the other seems objective. So you've got, I see how you could measure somehow pruritus, but if you're looking at something like fatigue, I mean, people have busy lives. There's busy professionals, there's people with small children, there's life changes, there's a lot of things going on. How do you measure fatigue?
Ben, you can speak to this too, but generally speaking, in the clinical trial realm, when we are measuring these types of outcomes, we measure them with what we refer to as patient-reported outcomes, or PROs. And so we ask the particular participant a series of questions. And as they answer those questions, we use that to determine their level of fatigue or pruritus, and we record that. That person's, what we refer to as their baseline, as when they come into the trial, is what we compare them to as they participate in the trial. And so their own answers to those questions are what we compare them back to. And so as they experience those life experiences, whether it's fatigue or whatever it might be that affect those things, they influence them. The reason why we have a placebo control and the reason why we enroll 350 patients in a PSC trial is so that we kind of filter out some of that noise. In addition to that, the patient-reported outcomes are what they call validated. So the particular questions that we ask to try to get at that question of fatigue or pruritus are studied in a way that validates that question, so that it's not just asking, do you feel fatigue, but it's asking several questions that, taken as a constellation, get at that particular issue in a way that allows us to measure it in a manner that we can then rely upon to determine whether someone is fatigued in a sense that we can measure it objectively through the course of the trial. Ben, do you want to take a stab at that?
I think we have a dedicated session right after this break on clinical trials. I think we will explore more of those questions. I would like to close session 1 and let everybody have a coffee break. We'll be back here at 10:55. Thank you for our speakers for such a wonderful program so far, and we will see everybody back here in about 10 minutes. Thank you.